Welcome to Muscle Part 2. We're now talking about the sliding filament theory, continuing on from the previous one. So we're thinking about how it is that muscle shortens. I started by telling you that there was differing overlap of actin and myosin, and that is what was going to allow for the change in length. Well, how so? So we would need these two layers of actin and myosin that somewhat overlap, and they have to be able to attach to each other. So if they can attach to each other, they can actually move and slide over each other in such a way that would allow the shortening of the muscle. So crucial is to know that the filaments never shorten. I'm going to say it again, the filaments never shorten. It's the overlap of these filaments, like two rulers that slightly overlap, and you just slide them over each other. Then the distance from the end of one ruler to the other is going to shorten if you slide them so that they overlap. In a diagram, you would go from this top one, where there's overlap of pink and purple, or actin and myosin, and it sits there, but if I can attach these and make them slide, then I would go from this diagram to this diagram, where these arrows, as you see, are moving in. This is the direction, and the overlap has become greater. And here you're seeing the myosin reaching up to the actin. And it does that, and it will be able to then grab the actin and move it and release it, and grab it and move it and release it. And in that way, make the actin slide over the myosin and reduce. If I extended this line here, this is how long this would be, this is how long the end of the other one would be, and you see that we contracted this muscle. If you were to look at it in a photograph, it would look like this, where you have that Z line. If I extend it here, oops, that's hard to draw with this, there, and over here, that was larger, and it's going to close in on itself. So let's go back to actin and myosin and see the mechanism for this to happen. Myosin attaches to the actin and pulls the actin toward it, causing the sliding of the filaments over each other. So it's the action of the myosin that has the capability of reaching up to the actin and pulling it. Let's look how. Let's look first at the components of the thin filament of actin. Actin is called F-actin or filamentous. So the F is for filamentous. Why is that? Well, because it looks like a filament. And these, long filaments is where it got its name. But really the filament, when you look at it more closely, you see that it's not just one strand, but it's two strands of a globular G-actin. Globular means like balloon-like, or like little balls, like globules. So you're seeing that it's one strand here, another strand under it, that coils around like a braid almost. Each one of those are globules or G protein. This would be the myosin here, and the Z line. Okay. What else do we need to look at in the protein actin? So here we can see that we also have something called troponin. Troponin, then, this is the globular actin, correct? Sitting on one of those globules would be this molecule of troponin. What it will do is it will receive the calcium that is needed for that contraction. So it's sitting there waiting for calcium to arrive. But let's continue with the components. So if that's troponin, what else do we have on this double, braid-like structure? we have tropomyosin. So tropomyosin then is this green ribbon. So the green ribbon is tropomyosin. This is troponin that's going to receive calcium. And what is the function of tropomyosin? Tropomyosin blocks here and here and here the attachment site for myosin. So if I keep this actin blocked, then the myosin cannot reach up and attach to it, and there's no contraction, and the muscle is relaxed. If I were able to remove this tropomyosin, then it would be ready to receive the myosin and contract. Okay, we'll continue with the parts first. What else? We're still talking about the thin filament. And we've looked at troponin. We've looked at tropomyosin. And then there's another big protein here in the middle. Do you notice? That's separating these balls, and that's called nebulin. So it's building up all the parts that make up the thin filament. Actin, troponin, tropomyosin, nebulin, and one more, titin. So at the end of the Z line here, it's like a rubber band that attaches onto the next one, and that rubber band is called titin. So all these are going to be associated to that thin filament. You need to know all these and what they do. What's the thick filament look like? In purple here is the thick filament reaching up. Here you see too the thin filament. And it's often described like golf club heads. So this is a head that's going to reach up. In this case here, it can't because tropomyosin is blocking it. If I were to remove the blockage of tropomyosin, then the myosin head would come up and attach. Myosin heads have two binding sites. One is where it's going to bind to actin. We're going to call that the actin binding site. So right here. The other is a nucleotide binding site that's going to bind ATP. And why does it need that? Because in order to contract a muscle, you will need ATP. And it's going to release one of those phosphates to be able to use it in that mechanism of this golf club head moving that way and making the actin slide. Okay, actin binding site and a nucleotide binding site that's going to bind ATP. This myosin head is hinged. Here's the hinge. And it bends and straightens during contraction. You have videos that I have posted for you on this. So imagine it coming up, attaching, moving that way. And then it's going to come up behind it, attach, and move that way. And then come behind it, attach, and move that way. And in that way, it's going to make the actin slide. Easier to see on here, where you would have this coming up and making it slide in that direction. I'm sorry, it keeps changing the slide on me. Those titin and nebulin are huge proteins, and they are going to provide this elasticity and stability. So if you think of the nebulin in the middle there, it's a stabilizer. It's going to hold that globular actin together. Very important. And the titin is, I think of it as a rubber band here that attaches onto the end of the Z line so that when this is contracting, there's still something attached to the other end of the Z line. It contracts within Z to Z line, but something's holding on to it, and it's titin at the other end. As I'm telling you, titin is a huge protein. It's the largest known protein ever that we know. It has 34,000 amino acids. It's also the largest known word. And if you look at the video on A&P, I posted it for you with Crash Course, A&P. He talks about the largest word, because it goes on forever, the actual name of titin. And as I told you earlier, nebulin is going to bind and stabilize that actin. Okay. We're going to stop here and look at more slides on the mechanism in part 3. Thanks.